Hey guys, so in this video, I'd like to talk a little bit about the process of meiosis. Right? And I have today for you, and your great viewing pleasure, a PowerPoint presentation. So let us go through this. Oh, cool. Alrighty. So meiosis, as I'm sure you know, is a mechanism of cell division specifically for sexual reproduction. Right? It is different than mitosis in a few fundamental ways that result in a vastly different outcome. Many of the mechanisms utilized within meiosis are akin to the mechanisms utilized in mitosis. So much so that one would assume that meiosis is an offshoot of mitosis. Right? Mitosis came first, some modifications occurred, we got the ability to do meiosis, right, through the course of evolution. And so the function of meiosis is to generate daughter cells that have half the amount of DNA as the parent cell. Right? And when we say half of the amount of DNA, we're not taking any arbitrary half and sequestering it away into the gamete. We're taking a very specific kind of half. Right? And so this is easiest to talk about with an example. Right? As humans, we are diploid, meaning that we have two copies of each type of chromosome that we're in possession of. As humans, we have 23 different types of chromosomes. And so being diploid, we have two copies of those 23 types. Meiosis separates those copies into halves so that each gamete gets a full set of 23. They only get one copy of each of the 23, but they get each of the 23. Obviously, that's important because if you're trying to make a baby human, they need all 23 chromosomes, right? So the mechanisms of meiosis are what lead to this division of our homologous chromosomes, right? Being, again, diploid, we have two copies, so I got these homologous chromosomes. Same exact style of chromosome. They have the same structure overall. They possess the same genes. Okay, they probably have different alleles because one came from the mother, one came from the father. Okay, homologous chromosomes get separated. That's what meiosis is doing. Yeah, and so we got a cute little diagram down here. I don't know if you can see this tab that I'm moving. It was in the way. If you can't see it, then don't worry about it. If you can, then you saw it. So we have this diagram. We start with a parent cell. It has two of these chromosomes, right? Because it is diploid. This is just an organism that has one type of chromosome, diploid. We have S phase makes a copy of each. And so we have the sister chromatids attached. We line up the homologous chromosomes. They get separated through PMAT1. And so PMAT1 generates a haploid daughter cell. It now only has one type of this type of chromosome. It only has the blue, for instance, in this case. And this one has the pink one. Right. Oi! And then meiosis II, PMAT2, separates them further. So each gamete, each daughter cell gets one copy of that one chromosome. Okay. Onwards and downwards. Here we have a wonderful diagram of the same storyline, right? So this is a little bit more specific. And this is the generation of sperms and eggs. And now sperms and eggs are types of gametes. They're the types that we use. Right? So obviously if you're a male, you generate sperms. If you're a female, you generate eggs. Okay? One huge difference between spermatogenesis and oogenesis is that when you're making spermies, every meiotic cycle results in 4 full spermies. Whereas when you do meiotic cycling for making the egg, right, you get 4 daughter cells, but 3 of those daughter cells because this polar body doesn't get messed up, are called polar bodies, right? Oi. Anyways, you get 1 good egg at the end of the story, right? And so after PMAT1, you have a good cell that's going to become an egg. You got all the cytoplasm stuff, right? And then you got what's called a polar body, and it's basically trash. It got the DNA that was separated through meiosis I, but it didn't get any of the material to become a cell. So this is effectively trash. Right? Then that good one goes through meiosis II. One of them is a good one. That's going to be ultimately the egg that we make as a female. Well, if you're a female, you make this egg. And then you get another polar body, another piece of trash. Right? Bloop, get rid of it. So when you do meiosis as a female, you get one good egg. Even though you had the material to make 4 daughter cells, you make one good one. Okay? And the spermies, you get 4 spermies. Yeah. So just a quick run through of the milestone steps of meiosis, right? So these are some of the important phases that are occurring along the way. And so at the very beginning of meiosis, we have prophase I, right? So PMAT, prophase, metaphase, anaphase, telophase. Prophase I is the first prophase that we're going to be doing. Within prophase I, there are developed these axial elements, which are protein structures that are going to help the DNA condense into its appropriate form and also allow for these sister chromatids, which are the 2 copies of the same DNA, to stick together. Okay. And then, right before we move into metaphase, when we want the homologous chromosomes to align, okay, this axial element plays a big role in that because it's kind of like a structural component. It's like a big bar that the DNA is attached to. And when I want the 2 homologous chromosomes to come together, the 2 bars can connect. And we'll look at the mechanism of connecting in a couple slides. Yeah. But one of the first things that has to occur before any of this meiosis can happen is this development of these axial elements. Okay. Then we have the alignment of the homologous chromosomes during prometaphase I. Okay. So this is a vastly different setup than we see in mitosis. Right? The fact that the homologous chromosomes align with one another, okay, along that midline of the cell, is very important for the way that the separation is about to occur in anaphase. Right? And so the homologous chromosomes with their sister chromatids respectively, right, get pulled apart together. Okay? And then they get lined up on the edges. Whereas in mitosis, you just had the sister chromatids get pulled apart and put to each side. Right? And so because we're moving the entire homologous chromosome set like this, okay, that's what's allowing for that generation of a haploid daughter cell. Because I'm only getting 1 copy of each type of chromosome going in either direction. Right? So this is chromosome type 1, A version, B version. A gets over here, B gets over here, and that's it. This side only ever is going to have A, this side's only going to have B, haploid. Right? Where in mitosis, you have the A version getting separated from the A version, so you get an A version on both sides, and you have the B version and the B version getting separated. You have the A and the B on both sides. Diploid. Okay? So this is very important. You have to understand that homologous chromosomes are lining up together along the midline in meiosis. All right. That also allows for the opportunity for crossover to happen. Right? The homologous chromosomes have the same genes on them. Right? And so there's a moment where they kind of be friends, right? And they trade a little bit of genetic information. Awesome, cool stuff. Gonna look at that in a couple slides. Okay, very important for creating genetic diversity. Okay. Continuing onwards, right? As I was just saying, when the homologous chromosomes separate, those sister chromatids are staying together, right? And so that's important during anaphase because now you have 2 copies of that same type on either side. Okay. The telophase I is actually going to create those 2 daughter cells that are now haploid. So as soon as meiosis I is done, I already have haploid cells. These haploid cells have 2 copies of each chromosome, but those 2 copies are the same as each other, right? So they're haploid. The daughter cells are half of the ploidy of whatever the parent cell is. And so we typically talk in terms of diploid because we're diploid. A lot of animals are diploid, a lot of plants are diploid, but there's a bunch of plants and a bunch of organisms that aren't diploid. You know, you can have multiploidy, like fourploid, right? And so if you're 4N, which is the connotation for ploidy, your daughter cells here are going to be 2N. We're 2N, so our daughter cells are 1N. If it was 122N, why did I do this to myself? I'm not going to do that. If it's 120N, it'd be 60N. Therefore, 122 would be 61N. Maths. Good job, Mr. Lavender. Cool? All right. So we get them separated. Cool. That's the end of meiosis 1. That's the end of PMAT 1. PMAT 2 is less interesting. You don't have any of the crossover storyline. You don't have to worry about the homologous chromosomes lining up. It's more akin to kind of like mitosis, but with less chromosomes floating around. Right? And so the only interesting thing that happens really is that during Anaphase II, now I'm taking those sister chromatids and I'm separating them. Right? And then at the end of this Telophase II, I now have my haploid gametes. Full haploid gametes. They have one copy of each chromosome, ready to make babies. Okay. So a very important principle of meiosis in the way that it separates the genetic information is the concept of independent assortment. The whole point of doing sexual reproduction, because it takes a lot of work, right? It's very energy inefficient. You got to find partners. You got to do the whole mating stuff, right? So sexual reproduction is not a very energy efficient system, but there's a huge payoff in the amount of genetic diversity that is generated. And so independent assortment is one of the steps that lend itself very well to creating genetic diversity through sexual reproduction. Right? And so the notion is that when you line up the homologous chromosomes during prometaphase I, you have the maternal version and you have the paternal version. So if you imagine the left side and the right side. There's no rhyme or reason to the way in which those sets are organized, right? So it's not like all the paternal end up here and all the maternal end up here. Or like maternal, paternal, maternal, paternal. It can be in any arrangement. It happens randomly, right? And so depending on the day, depending on the hour, depending on what side you're laying on while this happens, not really, but maybe, right? You can have different arrangements of these homologous chromosomes along the midline. And so this diagram demonstrates that. So in possibility 1, we have the paternal lined up on one side and the maternal lined up on one side. Right? And so then when you do meiosis 1, you get this setup, and then at the end of meiosis 2, you get this setup. Right? So these guys have both paternals. Both paternals, both maternals, both maternals. Whereas in this possibility, right, different day, okay, we had the paternal for this one, but the maternal for that one. We had the maternal for this one, but the paternal for this one. Right? So I have a different setup. And ultimately, that generates different gametes. Right? And so instead of being paternal-paternal, I now have paternal-maternal. I have a different set of genes, or rather, a different set of alleles. Same genes, different alleles. Okay? And so depending on how many types of chromosomes you have, independent assortment can create a lot of diversity. Right? This is just an example with 2 types. Diversity is not huge. Okay? But you go to 4 types, it's a lot bigger. It's an exponential increase. And then we have 23 types. Right? So if you think about the exponential increase of 23 over 2, that's a huge number of possibilities just through independent assortment. So this is huge in generating genetic diversity. This is why all my spermies are different from one another. Right? If I got spermie 1 and spermie 2, and they're like, hey, buddy, do you like ginger? This guy's like, nah, man, I don't like ginger. Because, maybe my mom's in DNA said, I don't like ginger. My dad's DNA was like, yeah, I love ginger. And so they're different. Right? Very important. Yeah. The other important thing for creating genetic diversity in meiosis is the process of crossover. So where independent assortment was dealing with the randomness of the entire chromosome arrangement, crossover deals with smaller sections of each chromosome. Crossover can create almost infinite genetic diversity because the crossover can occur at different spots on the same chromosome at different times. And so what happens is during prometaphase I, when the homologous chromosomes are aligned with one another, they overlap. So you can see here, we have sister chromatid 1, sister chromatid 2, 1, 2. The ones on the inside overlap. They touch, they physically connect. And in that connection, a process occurs that opens up the DNA, and the two exchange their DNA strands. They physically be like, oh, here's mine, here's yours, tradesies, no takebacks. Right? And so they trade their DNA. Actually physically swap the piece of their chromosome at that point. And so what I get at the end of crossover is 4 unique chromatids, right? And so this one is no longer the same as this one is no longer the same as this one. Okay? And again, that crossover can occur at several different spots within this chromosome. So it's never going to be the same 2 times. So you get a huge amount of diversity through this process of crossover. Let me move my beautiful face down a little bit. All right. So synapsis is the term for when the homologous chromosomes are aligned. Okay. Synapsis. The chiasma is the spot where the 2 homologous chromosomes are overlapping. It's called a chiasma. Oh, look at that. I wrote it right here. It's the spot where they're physically linked. The crossover is occurring at the chiasma. So we have our pre-crossover homologous chromosomes during synapsis. They're aligned. You have the moment where the chiasma is generated. They're touching, they're exchanging information. And you have your post-crossover recombinant chromosomes here, where they've traded their genetic information, right? These pieces swapped. So, if you recall back to when I talked about the axial elements, so the axial elements run along the bottom side here. You can't really see it great in this image because it's black on black. Okay, but it's kind of like a bar. Just imagine there'd be a black bar right here on each side. Okay. The squiggly lines are the DNA, the chromosomes. Okay. The different tones represent the different sister chromatids. Right? So blue is one set, and they're the same. They're both blue. And red is one set, so the red and the pink are sister chromatids. Yeah. This green structure here is a protein complex called the Synaptonemal Complex. Okay. And it's literally just a big protein filament thing that runs between the 2 homologous chromosomes and physically holds them together, right? It's like a big string. If you sewed a big string between the axial elements, between the bars, right? Tie them together, hold them in place. Yeah. This complex will stay here throughout all of prometaphase, okay? And once metaphase fully begins, then it starts to fall apart and disappear. Okay? But for now, there's this protein complex that's holding together the homologous chromosomes. Okay? It allows for these tiny regions right here to connect. This would be the chiasma, and that's holding together, or rather, that is where they're exchanging the DNA. Okay? So this guy right here, synaptonemal, synapse, right, is this big green protein thingy right here, tying together the homologous chromosomes. Okay? On the DNA itself, right, there are regions that have certain coding patterns that give it a specific shape that promote crossover. Right? This is where the 2 things can exchange. They kind of stick to each other. Okay? They're called chiasmata. These chiasmata, these regions of DNA, are where the chiasma are formed. Okay? It's where crossover happens. Okay? And so I was exaggerating a bit earlier when I said that it can happen anywhere along the DNA. Right? There are specific regions that it can occur, but there are many variations of these regions throughout. Right? So it's going to be various different spots that crossover can happen. Okay? And so the chiasmata will connect, you form a chiasma, you can have crossover. You can have multiple chiasma on one set of homologous chromosome per meiosis. So you can have more than one crossover event. More DNA mix up, more genetic variation, awesome. Okay. And so once the chiasma are formed and connected, you have a protein that is named Spo11 or Spo11. I've honestly literally never heard it said out loud, so I don't know if it's Spo11 or Spo11. I'm going to go with Spo11 because it sounds cooler. I like the way it rolls off the tongue, the Spo11. Okay. And so the Spo11 is the protein that's responsible for breaking the bonds of the DNA at that chiasma. Okay? And so it opens up the DNA, clicks them off. Okay? And once the two DNA strands are open, because they are the same genes, right, where the chiasma is, they can switch. And when they click back together, they can click into that new chromosome set, that recombinant chromosome. Right? And so you have the two pieces, they break open, they swap, they click together, and they move apart. So you got some diagrams here that kind of show that. So here we have the Spo11 clicking onto the DNA and then breaking open it. Can I move this? Oh, what happened? I want to move this. There we are. Okay. So we have the Spo11 clicking on this DNA. Click, click, click. Cracks open the DNA in that spot. You can see that there's this opening here, right? It's kind of like an overlap. Okay. That overlap's important because where that overlap is, that's where the next one, when the other set, the homologous chromosome set, is broken open as well, they overlap and they click together, kind of like Legos. And here in this diagram, we see that happening. Right? And so you can see the short strand and the long strand where they overlap and they connect together. Right? See this little bendy, click, 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 and then you get your crossover here. So the dark one connected to the lighter one, and the dark one connected to the lighter one. Overlapping, connect, separate, recombinant chromosome. Here we have, again, a revisiting of the results of crossover. Why was my face not moving? Why is my face not moving? Here we go. All right. And now I'm sepia for some reason. And now I'm reversed for some reason. And now I'm full screen for some reason. And now I'm blue. Okay. All right. Well, I'm staying sepia. Anyways, I think this is the last slide, or at least very close to the last slide. The way that the chromosomes are moved in the process of meiosis. Right? And so there is the organelle, the centrosome. Right? And so the centrosome at the beginning of meiosis or mitosis really matures and develops. Right? And it reaches out in microtubules, which are like little strands. And so the centrosomes on the edges, they reach out their little strands into the middle, right? And they're floating around until they connect to one of the chromosomes. Okay? And so on these chromosomes, right, in the middle, where the sister chromatids are attached, there's a protein structure that's wrapped around them. That protein structure is called a kinetochore. That kinetochore serves as an anchoring point for the microtubule. So you got your kinetochore attached to your chromatids, okay, like this. The microtubule is like floating around, and like eventually, like, oh, it's like boom, pokes onto it. Kinetochore grabs it. Now it's locked in. It's good to go. Okay. Once all of the chromosomes are lined up in the middle appropriately, because now we're doing meiosis, the homologous chromosomes are lined up like this. Right, they're held together with that synaptonemal complex. Okay. They do their exchange, and now it's time for anaphase to pull them apart. The centrosomes spin, and they reel in the microtubules, and that pulls the chromosomes to the edges. Okay? And so these microtubules attached to the centrosomes are coordinating the physical movements of the chromosomes throughout these steps. Okay? And so it's like a big string on a spool. It's really simple in the physical sense, just like all biology things are really simple in the physical sense. Right? I mean, they can get complex in like the multitude of details, but conceptually, it's all just very straightforward, pulling them with strings. Any other more slides? That's it. Cool. Awesome. So we have meiosis. It's a two-step cell division geared towards making daughter cells that are half the ploidy, or whatever the parent cells are, so that they can be used in sexual reproduction. These steps of meiosis are geared towards creating genetic diversity. We achieve that through independent assortment and the process of crossover. Cool. Bye.